Good morning, all of you. You are welcome to this online event. This is actually a curtain-raiser event for our 17th Foundation Day, which is scheduled to be next Wednesday. And this occasion, I would request our director, Professor Neeraj Jain, to say a few words.
Yeah, let me just share my screen. Can you? I hope you can see that.
We can see it, sir. Just make it full.
Yeah. Yeah. Okay. Great. Yeah. So, okay. Good morning, everybody. I'm director of National Brain Research Center, and what you see on the screen right now is a panoramic view of National Brain Research Center, which is located in Manesar, about 40 kilometers from Delhi. You can, there is a link which is available on YouTube, and you can learn all about what NBRC does. There's a film that we have recently uploaded there. NBRC was, let me just go back because I think my slide didn't move on screen.
We can't see your slides actually. Yeah, now we can see.
We are on slide number 2.
Yeah, now you can. Yeah, now you can see slide number. I'll keep it like this. So, you know, as Professor Anirban said, NBRC was actually inaugurated, or I would say more appropriately, dedicated to the nation by the then President APJ Abdul Kalam, who was very interested in science, was very interested in talking to children all the time. And that day, every year, we celebrate as our Foundation Day. Normally, we have outreach activity, which culminates in the Foundation Day lecture. But in the morning, we have children from various schools in the neighborhood coming to NBRC to see what we do at NBRC through exhibits, interacting with scientists, interacting with students, going around, learning about our work, experiencing the thrill of the research that is there, including, which personally for me is the biggest thrill, is researching on the brain. Unfortunately, due to the COVID situation, we are not able to call children to spend half a day at NBRC campus. And you can see the last year in this particular slide, we had a quiz competition, and the winners of that quiz competition are being given prize there. So we decided we will do whatever we can under the circumstances. That's why we have uploaded that film about NBRC so that everybody can watch it. And in lieu of sort of this outreach activity that happens in the morning, we have organized a number of lectures, and today's lecture is by Professor Pankaj Seth. Professor Anirban will tell you more about him and his research work. And I will just thank you all for joining and welcome you to this first event for the NBRC Foundation Day celebrations this year. It will culminate on December 16th by a public lecture by Professor Joseph Rao Shekhar, which will be available on the YouTube link, which is given here. And I suggest that all of you join. That will be at 6:30 p.m. in the evening. This is bound to be an exciting event. I am so glad that Professor Pankaj has agreed to this lecture, and we continue to welcome any queries, any schools, anybody who would like to learn more about it, who would like any of our faculty to deliver at present a video lecture to the students. Professor Anirban, can you please go ahead and introduce Professor Pankaj now?
Thank you, Professor Jain, for the kind introduction about the NBRC creation and how NBRC has been created. So now I'll introduce Professor Pankaj Seth, I think it's one of the easiest introduction I have ever given, because I know him so well, so I don't have to Google or I don't have to see website. Even I can give it impromptu, almost virtually. So what Dr. Pankaj did, so he did his PhD from CDRI, Lucknow Central Drug Research Institute. Following that, he has moved to US for his postdoctoral training. And after second postdoctoral training with gene major in NIH, he moved to NBRC to start his lab. In last 17 years of his stay at NBRC, he has developed a group, vibrant group, working with the defined area of the neurotopic viruses, and most importantly, HIV and Zika. He has very elegantly showed how these viruses infect the neuronal stem cell, glial cell, and they're responsible for the death of neuron. And most importantly, he has developed an important stem cell platform, which very used by the investigators across NBRC, and not only NBRC, across different institutes in the country. And it's a very important tools which showed that how from the aborted fetus you can get the specific cell like astrocytes or oligodendrocytes or the neuron. This is the one part of his credential. Another most important thing I think I should mention about Pankaj is that he put enormous effort to make neuroscience popular across all the level. With his association with Indian Academy of Neuroscience, he tried to organize a session every year, and I know how he's very keen to introduce neuroscience and popular neuroscience at all levels, from school level to the college level to the medical college. He tried to go visit and tell them how NBRC is working and how the neuroscience is developing in this country and improving day by day. Without that, I would like to Dr. Pankaj Seth to give his talk for this on the occasion of the 17th Foundation Day of NBRC. Pankaj.
Yeah, thank you, Professor Jan, for giving me this opportunity to be giving the first talk in our, much awaited Foundation Day activities. As Professor Jan has mentioned, we do a lot on Foundation Day. Unfortunately, we are not able to do those things physically, so we are trying to reach out to the general public through virtual means. And thank you so much for one of the best friends at NBRC and my next door neighbor in terms of office space, Dr. Anirban, who's been very modest in saying all that and very generous for the generous introduction. Now coming to the topic which I've chosen today is also timely because, we all are worried about the COVID situation, the crisis, and everybody wants to know more about this virus. Now, viruses in general do affect brain function, and that is what I have thought that, I would tell you how some of the popular viruses in the past, infamous, I should say, like HIV and Zika, and now SARS-CoV-2, affect the brain functions. So we started our work. My lab is situated here, if you can see the cursor. And what this institute, a beautiful institute situated 40 kilometers from the Delhi airport at the place in Manesar, a very quiet, serene location, green as you can see, has a wonderful team of scientists and researchers who are trying to understand how healthy and diseased brain works. My lab is interested to understand how viruses affect human brain. So research as such in general is like putting pieces of puzzle together. And that is because, one scientist cannot, have the complete picture of what is happening in normal basic understanding of any function, be it brain, liver, any other vital organ, or in terms of diseases. So often it is not possible to do it alone. So you have to have collaborations, which we always do. All scientists at NBRC collaborate within themselves and as well as outside, as and when required. And it requires a lot of patience, passion, and sustained funding support. And I'm saying that because I know there will be a lot of student population who may be joining this lecture. So have patience, have passion, and of course, your guide, your supervisor, your director, and your head of the institutions will maintain a sustained funding support so you can do the research what you want and answer research questions. So, to find answers for a disease, usually there are two ways to go. So the scientists work from bench to bedside, as my friend and collaborator, Dr. Anirban Basu, has done a wonderful work to understand how JEV is affected, and whatever he learned in his lab, he applied that to find out and reduce the burden of mortality and to some extent morbidity of the kids who were infected with JEV infections, because he had done a lot of basic research with a compound called as minocycline, which was and repurposed that drug to solve the problem of JEV. In our lab, we have been actually going the other way mostly, because whatever symptoms we found noted from clinicians, from literature, and we tried to figure out what is going wrong in the brain, whether it is HIV or Zika, and try to find answers at the bench, because we have a wonderful tool which I'm going to just explain to you. Now, for doing research, whether it is basic or clinical, you have to have inquisitiveness. That is paramount, students. You have to always you have to be inquisitive about finding. And I'm very glad to share with you that, the group, what we have small, but a very dedicated group of researchers at National Brain Research Center, which are 24/7, they're thinking and they're inquisitive to understand how healthy and diseased brain works. Now, if I said you should be inquisitive. So the first thing which should come as a researcher in your mind should be, if the topic is how viruses affect human brain, the very first thing which should come into your mind is the first question: can the viruses reach brain? Despite the fact that the brain is protected by various ways, can still the virus reach the brain? Because this question is first question, because if it are affecting the human brain, the viruses have to reach the brain first. And the second question, or a logical question which should come to your mind as a researcher, is if it reaches there, how does it reach there? And what are the mechanisms the virus follows or the pathogen follows to get into the brain and cause the effects? And thirdly, what cell type? Brain is made up of a lot of cells. It is made of glial cells, neuronal cells, and other cells. So what type of cell is affected? What does the choice of the pathogen is? What decides the cell tropism? In other words, the viruses liking any particular cell type. Some of the viruses affect microglial cells more than neurons. So what decides that? How does pathogen know which cell I should infect? And they have the tropism for. And then when they reach a cell, whether it's astrocytes or neuron, what are the mechanisms and how do they take over the machinery of that particular cell, whether it's astrocyte or neuron, and they utilize that for their own benefit. I'm talking about viruses here. Now, the lab has been invested to understand two major viruses. Both have been said to be neurotropic. In other words, they have tropism or liking for brain cells. We all know that HIV causes AIDS, and usually it is more prominent after birth. It crosses the blood-brain barrier. It reaches to the brain and causes HIV-associated neurocognitive disorders. Zika, on the other hand, is another different class of virus altogether, but it starts its effect on brain in utero before the kid is born. And that is why when mothers are infected in their first trimester of pregnancy, this virus makes its way through the placenta barrier, crosses the placenta barrier, unlike HIV, which does so only after birth. This one starts working much earlier. And that is why when the kids, 7 to 8% of the children, which are infants, which are born with such mothers who are infected in their first trimester of pregnancy, end up in having microcephaly. Micro is small, cephaly is head. So small head size is what Zika virus results in. Okay. Many kids also, women who are infected also have stillbirth. So there's a lot of mortality which is connected to Zika virus. But fortunately, the population is now more or less developed herd immunity, and that is why we don't see that many microcephaly nowadays. And both have been studied in our lab by using primary cultures of human neurofetal brain-derived neural stem cells, which I'm going to touch upon in subsequent slides. Now let's start with HIV first. So if you could see this animation, what this animation is going to show you how the virus traffics into the brain. Now, HIV usually is said to be a disease of immune system, and that is why AIDS is developed, which is acquired immunodeficiency syndrome, where the T cells go down and the immune cells go down, And that is when the opportunistic infections come in and the person succumbs if he's not treated well. Now, in that process, much before the AIDS develops, the virus, HIV virus, traffics into the brain, as is shown in this animation. So you're seeing at the cross section of the virus, this is the GP120 studs which are there. They will get into the blood by various ways. And since the blood traffics to all vital organs, including brain, it gets into the microvasculature. Now you're seeing a section, cross section of one of the blood vessels in the brain. And as you can see, this is the endothelial cell lining, as is there in all blood vessels. In the brain, they form an important selectively permeable natural barrier which prevents the blood and bad things of the blood to come into the brain. Only the good things are allowed. And what you're seeing in this frame is one of the monocyte blood cell, which has these white dots, which are viral particles. And this is one small virus, RBCs, another monocyte, which is not infected, but another viral particles. Now, the beauty of this selectively permeable barrier is it doesn't allow cells or particles based on their size. It allows allows the access of blood cells into the brain based on the requirement of the brain. Now, obviously, viruses are not required in the brain, so they don't get access through the blood-brain barrier. This is the blood-brain barrier, which is formed by endothelial cells, and they have support of several astrocytes, which give the blood-brain barrier. Now, but the monocyte is a cell type which is allowed through the blood-brain barrier, and you will see that in the animation. Why monocytes, which is much bigger in size than the viral particle itself, is because monocytes go as surveillance cells in the parenchyma, and then they can help to provide the limited immune system. So if we continue with the animation again, and I bring it to this frame again, you will see that how the monocytes will get access through the blood-brain barrier, endothelial cells. And you see here a monocyte coming in, which is infected, loaded with the virus, comes here, gets access into the brain parenchyma. And this is how viruses get into the brain. They dock to the CD4 receptor. It is taken inside, and the nucleic acid is released in the first host cell. This is how infection occurs of HIV in brain cells. Now, once they get into the brain cell, this is what the animation showed you. There are two ways by which they can affect the neuron. Interestingly, they do not infect the neurons, but neurons are being affected. They're affected because by two ways. Number one, when the virus gets into the brain, the viral proteins which are there on the surface of the virus, they're also shed by the virus. One of the protein which is shed is the GP120, the protein which the virus uses to dock to the CD4 receptor to get access into the brain. They're also shed. The other protein which the virus makes is the TAT protein, which is the transactivator or transcription of the TAT protein. And that protein and GP120 are highly neurotoxic to the neuron. So this is one of the direct mechanisms by which the viral proteins affect the neuronal cells. And the neurons, which are the functional blocks or functional cell of the brain, are affected. The other way is that they affect the microglial cells where they multiply, the viruses multiply. And these microglial cells also give more viral particles, induce a lot of cytokines and chemokines, and then affect the function of one of the most abundant cell type in the brain is the astrocyte. Now, astrocytes provide all the good stuff which is required for the neurons. Astrocytes provide growth factors, they provide trophic factors for the neuron, they take away the excess of glutamate, and so they uptake the excess of glutamate or the neurotransmitter so that the neurons do not get excitotoxicity because of that neurotransmitter glutamate. But if these astrocytes are affected because of the virus, they stop doing those two important functions. One is to provide growth factors to the neuron. Second is to uptake the excess of glutamate. And that results in cell death of the neurons, which by apoptosis process. So this is the indirect pathway. So this is how the HIV causes a neuronal damage. Now, because there's neuronal damage occurring in the cell, it started causing neurological problems in AIDS patients. And that is studied by a field called as neuro-AIDS, which is neurological deficits or compromise of cognitive and motor function in AIDS patients. Now, the field is trying to understand how HIV and viral proteins affect brain cells, and we are also trying to do that. Now, one of the major challenges in the neuro-AIDS field is because this is a human-specific virus, there are no good animal models. And you can't work with humans directly, so you have to have the brain tissue of HIV/AIDS patients from autopsy or postmortem samples, and that is available in very limited quantity. And usually, autopsies are done at the end stage of the disease. So there was a great need for forming a cell culture system of human origin, and that is where the right of choice of right model system is very, very important. And we at NBRC tried to establish that. And the thing which we established is very interesting. And this cell culture model is primary cell culture model derived from fetal brains of aborted fetuses, following all ethical protocols and informed consents. We get the brain samples from local hospitals of 8 to 16 weeks. We do undergo a lot of processing, which I'm not going to go into detail. This is all published. If you have any questions, you can get back to me on my email, and we'll be happy to share with you those things. So the beauty of this fetal brain-derived neural stem cell model is that these cells grow as monolayer cells. They can grow as a layer on plastic, or they can grow as suspension culture as neurospheres. And these stem cells, because as other stem cells have high proliferative ability and show all the markers which these neural brain stem cells should show. So they have SOX2, they have nestin, they are Ki67 positive, indicating their high proliferative ability. Using this model system, we have published a lot of papers understanding how viruses affect brain cells and other disorders as well. Now, I told you that they can grow as suspension cultures and form the neurospheres. If you look at this animation, this shows in time-lapse microscopy how one neural stem cells divides into 2, 4, 8, 16, and after 48 to 72 hours, it forms this beautiful one cell mix of a neurosphere, which can actually then be used as a model system as well. If you measure the diameter from here to here, you can understand what was the proliferation rate and how big the neurospheres are formed with time. You can also make them single cell and count them and figure out from one cell how many cells made this neurosphere. Because they are stem cells, they also have ability to differentiate and form new neurons. We also try to do that and understand if the viruses are affecting the proliferative ability, as you saw in the previous slide, or they affect the ability of the stem cells to make new neurons or the process of neurogenesis. So this model system gives us various ways by which we can study viruses or any pathogen for that matter. We have used it to understand HIV and Zika virus. So we use HIV and Zika virus viral proteins on neural stem cells. We also study how astrocyte formation is affected or how neuron formation is affected. We work with viruses or viral proteins on astrocytes, on neurons, and also as co-culture of astrocytes and neurons, because in our brain, each neuron is surrounded by a lot of astrocytes, and we have 100 billion neurons and many more astrocytes around them. Okay, so very quickly, now I'll tell you what is the potential of this human neural stem cell model. It can study the effect of HIV on stemness of these NSCs. It can study the effect of HIV on neurogenesis, on astrocytes, on astrocyte-mediated neuronal damage, as well as on effect of Zika viral proteins on neural stem cells. Now, whether stem cells are susceptible to HIV infection, this was proven by a very good study from a very simple study done in NIH, where they had shown for the first time that HIV is present also in the neurodegenerative cells. But coming to the research questions now, if it is present in neural stem cells, how does virus or its viral proteins affect the function of the neural stem cells? This is what we did. And the first thing what we found was when you have the TAT protein put on the cultures of neural stem cells, you see what happens to the markers of the neural stem cells. So if this is without TAT, this is with TAT. And what you see over here is that if you have TAT in the culture system, you see that some of the green cells are totally, that stain of the green is gone. What is that green stain? it was staining for cyclin D1, which is one of the cell cycle regulator. So what will that tell you? If you have the viral protein TAT of HIV taken in concentrations which is present in the CSF of the patients of AIDS patients, you can actually reduce the cyclin D1 staining, which means that the ability of the cell, neural stem cell to proliferate is inhibited, which is also seen with decrease in the Ki67 marker. Now, the other ability of the neural stem cells is the process of differentiation and process of neurogenesis. Now, if you go down here, you will see that, in day 1, day 3, day 5, you see increase in number of green cells over here. Now, this increase in green cells is indicating formation of new neurons if you do not have TAT. But if you have TAT protein in 25, 50, and 100 nanogram per ml concentration, you will see that the number of green cells is down. Total number of cells as indicated by blue dots or blue stains is also down, indicating that TAT in increasing concentration can affect the differentiation rate and the ability of neural stem cells to form new neurons. So by various ways, this is how we have understood at least part of what HIV does to the neural stem cells. So the other important thing is that you have to validate this whatever you found in the Petri dishes. Okay, so this in vitro work, whatever we do with cell culture, was also validated in brain sections from postmortem brain sections from Nimhan Brain Bank in Bangalore. This is a place which has brains of normal patients, normal people, as well as diseased people. They have for most of the neurological disorders and neuroinfections. They also had 40 brains or 140 brains of HIV AIDS patients. So we requested that to have it. And what we found is that the nestin, which is one of the marker for neural stem cells, it is found that the brown stain is very less in HIV cases. What does that tell you? That tells you that the pool of the neural stem cells is low in HIV/AIDS patients. How does that affect? That affects because whatever ability of brain was there naturally available and the process of neurogenesis could have happened cannot happen because the overall number of cells or nestin positive cells or neural stem cells is less in HIV cases. We've also figured out that what are the consequences of this decrease or the presence of HIV in the brain is it affects the proliferation rate through a factor called as TRIM32. Now, this is a stem cell fate determinant. Now, I'm not going to go into much details because I know this is a diverse audience, but I just want to tell you that the presence of TRIM, whether it is in the cytoplasm or in the nucleus, and its concentration in control cells versus HIV, you are seeing over here that in HIV cases, in the brains of HIV, the TRIM32 is more, but it is mostly in the nucleus, as it can see in these rounded cells. Cytoplasm is outside the nucleus, as is shown in here. So in the control cells, you have this TRIM32 outside. In HIV, you have it in the nucleus, and that is one of the reasons why viruses do not allow these neural stem cells to propagate. We have proven that. We have found out the microRNA also, which are important for these things. And I'll just bring you to this, our understanding of how HIV is working on neural stem cells through TRIM32 with this slide, worked out by one of the very talented students, Meher Fatima, who showed very elegantly that the TRIM32 modulates the proliferation. Now, continuing further, as I said, each neuron is dependent on the astrocytic function. We have published several reviews on how astrocytes are important, and there are many other reviews by others who very clearly indicate that astrocytes support neurons in various ways, and neurons actually cannot function in the absence of astrocytes. So that means that the astrocytes and neurons are in constant dynamic crosstalk and are talking to each other to understand what is required by the neuron, and astrocytes provides that. Now, if that crosstalk is being affected by the presence of the viral proteins or the virus itself, it is going to have an effect on the neuronal functions, and that's how the cognitive functions may also be altered. So we try to simulate the structure we have in the brain in terms of astrocyte neuron relationship. So what you see in this light microscopy picture is you see one neuron indicated by N over here, and you see several astrocytes which are surrounding one neuron. So this is how actually one neuron is surrounded by astrocytes. Now, why are astrocytes surrounding neurons? They're surrounding neurons to have a crosstalk, which is shown much better in this stained picture of neuron shown in green, which is trying to extend its processes to the neighboring red cells, which are astrocytes. And these astrocytes and neurons crosstalk at these junctions, which are formed between neurons and astrocytes. And these can actually be affected if you throw in this culture the tag protein. You will see that the neuron, which was connected with astrocyte in red, the green cell, actually retracts itself and forms this bead-like appearance. This is one of the first things which is showing that the astrocyte neuronal connections are broken because the viral protein, this because it is toxic, and this is because the cell which was actually helping the neuron starts making certain things which are not good for the neurons. So neurons disconnect from the astrocytes. This is what I would like to tell you. And we have worked out different mechanisms how that is happening. What are the receptors involved in the glial cells which are harming the neuron? We have also figured out that there are certain microRNAs which are regulating the astrocytic mediated neuronal death in HIV. And very recently, Priyanka has shown of a Delhi University student who joined us, has shown that indeed there's one other thing, mortallin, which also helps in reducing. So we are getting closer to understand how viruses, particularly HIV, is affecting the brain cells, our brain functions. Now, coming very quickly, in 5 minutes, I'll show you how Zika virus affects the brain cells and how we have understood that. Now, you would have seen this picture. This is the normal head size of the kid which is born. This is with severe microcephaly. Micro is small, cephaly is head. So the small head size of the kids which are born if the mother was infected at the first trimester of pregnancy. Now, does it really happen? Yes, it does. Because these are the pictures of some of the microcephalic kids. As you can see, many of them die during birth. Some which are born have abnormal small head size. So their life is stunted, their mental abilities are stunted, and they have a very poor life outcome later on as well. So this is something which is to be worried. And the association of Zika virus with microcephaly was found by WHO way back in 2015, 2016, when during Brazilian Olympics, around that time, it was found that there were a lot of countries which saw a sudden spurt of Zika virus outbreaks, as well as Zika virus associated microcephaly. And this is how association was brought between these two things. So then people started researching on it, and many people showed in various ways that Zika virus can indeed affect the neural progenitor cells. In cell culture, in mice model, and in human system, people then later showed that the Zika virus impairs the growth of the neurospheres, the animation which I showed you, something similar, and brain organoids, mini brains as we call them. Now, this research was in progress, but none of these papers were indicating what are the mechanisms by which the Zika virus or its viral proteins, or what component of the viral virus is affecting the neural stem cells. That is where NBRC researchers came along, and we started researching on very simple experiments. All we did was we had these neural stem cells in our lab. We took structural proteins and non-structural proteins of the virus, Zika virus this time. And structural proteins, one was the E protein, non-structural proteins were the other proteins of the virus, and we started looking at them and their ability to affect the neural stem cells. What we found was very interesting, that while the Zika virus E protein was affecting the proliferative ability of these neural stem cells the most, other structural proteins were not affecting that much. So we went ahead, did our neurosphere assay in one of the Petri plates where we formed the neurospheres. We formed the big vector control without E protein, the envelope protein of the virus, the structural protein of the virus, Zika virus. And when we had the structural protein and in the neural stem cells, and then neurospheres were formed, we found there were fewer spheres and smaller spheres as compared to the vector control. So we saw an increase in the small sphere size, which indicated that the proliferative ability of these cells was affected by E protein. We also did differentiation, and we found that the new neurons which were being formed, or the process of neurogenesis, which was here in normal cell, new neurons were being formed from stem cells. They were branched, they were migrating. In E protein, or the envelope protein of the Zika virus, they were not doing so. So the neurogenesis process was also affected in E protein, suggesting that the migration and the formation of new neurons was affected by the E protein. So this is how we figured out that what component it is, the envelope of the virus which is affecting the neural stem cells. And since it is all happening during development, in early stages of development, that is why perhaps the neural stem cells are not able to proliferate, not able to differentiate, and not able to form the brain or the head size. And that is why perhaps these infants were born with abnormally small heads in Zika viral infections. So we did a lot of other microRNA studies, which I'm not going to go into detail. We took neural stem cells, transfected them with E protein, and then looked for differentially expressed microRNAs. What we found was that microRNAs which were differentially expressed were important for proliferation and differentiation of neural stem cells. Then we did one final experiment, which was by trying to simulate that in vivo conditions. So we had mouse model system which mouse were during development, the pups in utero were zapped or were electroporated with E protein. They were basically, it was seen that the virus is successfully, or the vector and the expressing vectors were there in the brain because they were labeled with EGFP. And what you see over here is this is the layer of the developing brain. So without E protein, you can see a lot of red cells, which are indicating SOX2, one of the marker of neural stem cells. EGFP concentration is more or less similar, which is indicating that the transfections were more or less same. In the pups which are transfected with E protein in utero, you see that the layer size is smaller, so the brain development is smaller. And the number of red cells is also small, indicating that number of stem cells are less, which is indicating the proliferation is less. If you look in this other slide also, you see these red cells. These are proliferative marker Ki67. And if you see in the E protein side, there's very fewer cells over here, indicating again that the proliferative ability of these cells is affected by E protein. So now we understood how two viruses, HIV and Zika, are affecting the human brain cell functions, and perhaps giving the understanding at the cellular and molecular level how brain functions are affected in HIV-infected individuals or Zika virus-infected individuals. So Reshma published this paper based, I didn't tell you all the details, but the major findings are that there's certain microRNAs which are upregulated. They are basically important for downregulation of Notch2 and PAX3, which are important for neural stem cell function and neurogenesis. Now, in last 3 slides, I will just show you very quickly in less than 5 minutes that, we are now focusing on understanding the cellular and molecular mechanisms of the SARS-CoV-2, the causative agent of the COVID pandemic. SARS-CoV-2 also causes neuropathogenesis, and we are trying at NBRC to understand how does it do so, so we can perhaps talk to the clinicians and tell them, perhaps try this or that and prevent the neurological complications in COVID patients. Now, does SARS-CoV-2 affect the brain? Well, that knowledge or that basic understanding comes from earlier paper from SARS, not SARS novel or SARS-CoV-2, but SARS was shown. Stanley Perman, who's on our board, external board as well, had shown earlier that it has ability to affect cause neuronal death. And there are several other papers which in past have shown that there's neuroinvasion happens in respiratory coronaviruses. So now, interestingly, there's now mounting evidence which is coming up that if you look at the postmortem samples of some patients, and indeed, I know most of you are now so well aware of the coronavirus symptoms. So you would know that there's a lot of headache in these COVID patients. Even people, when they survive and they're recovered, some of them continue to have headache. Many of them are also having problems of recall of memory. Some of them are having confusion, especially during active infection of SARS-CoV-2 or COVID. Many people lose their sense of smell, sense of taste. More people are having stroke. So all this is indicating that this virus has effect on brain cells and has neurological complications. Now, in a review, which was written by one of the students from our lab at NBRC, she has shown that what are the possible routes of entry of the SARS-CoV-2. This to be proven, but the possible route of infection, because it involves the olfactory or the cells which are important for smell, these viruses from the mucosal membrane can get into the olfactory nerve fibers and can reach into the brain, because the olfactory nerve fibers go and have connections with the brain directly. Now, from the blood-brain barrier, as you saw in the case of HIV, this virus can also traffic through the blood-brain barrier into the brain. It has been shown to be affecting the glial cells in vitro. Now, there are electron microscopy studies in postmortem samples, and their various cells have been shown to harbor the virus. The virus gets into the brain. There's no question about it. Although there is debate about it, but more and more evidence are coming. So we are also trying to get some sections of autopsy of COVID patients and try to trace the virus if it is there in the brain, and what are the steps it follows to get into the brain. One of the symptoms is diarrhea in COVID patients, and so the enteric epithelial cell lining is also affected. And it is believed that from the gut also the virus can get into the intestine and at various levels. And since gut is supplied with good nervous system and there's a gut-brain connection also, there's a possibility of the virus entering into the brain also through the gut. Now, when we have the cytokine storm in the peripheral system in our blood, it opens the blood-brain barrier. That gives another way by which the brain cells gets infiltrated by inflammatory cells from the blood, and this can also cause damage to the brain cells, because inflammatory cells are usually not there in the brain, but they can get into the brain by opening up the blood-brain barrier when you have huge cytokine storms, such as inflammatory cytokines like IL-6 and others. So these are various ways by which virus can get into the brain. And these are various ways by which people are studying what are the consequences of the virus in the brain. So some people have used human cortical organoids, various ones, cortical, hippocampal, hypothalamic, midbrain, and others are working on cellular level. We at NBRC are working at both the levels, trying to understand what viral proteins of that virus, SARS-CoV-2 or the novel coronavirus, what functions and what cell types are being affected. So the potential of human neural stem cells is immense. We have worked on HIV, Zika, and now we are employing the same model systems to understand SARS-CoV-2. And we have developed a platform, a cell type of neural stem cells, which are monolayer cells, suspension cultures, astrocyte neuron interaction, iPSCs, brain organoids we are developing now, and so on. So with that, I'm very grateful and lucky to have very talented students. The work which I presented today is mostly of these 3 graduate students, PhD students who came from various backgrounds to NBRC, and we could do this wonderful science. This is my group, and this group keeps changing. Many people come in, they go out after graduation, new students join, summer students join. And I'm very thankful to these very talented students. I'm grateful to the civil hospital doctors who have helped. And of course, nothing happens without money. And the funding for research came from various sources, including NBRC core funds. With that, thank you so much for your attention. And I hope this was useful to all. Thank you.
Pankaj?
Yes, sir.
So thank you so much for this wonderful and lucid talk. We have one question in the chat box.
Okay.
How HIV actually affect the memory? Because if it infect the neural stem cell, maybe there are some neural stem cell in the hippocampus also. So they affect memory that way? Because I think that's one of your first paper I remember in 2007 or 8. I think that's the probably you have to explain.
Yeah. So thanks for that question, Ankush. I don't know if you were able to follow the questions at YouTube. All of you can write to me on my email ID, which is there on the NBRC website, or you can Google me up. But for this question, the answer is that although HIV does not infect neurons, but the memory is affected. The memory is affected mainly because the support of the glial cells goes away and there's neuronal damage. And there's neuronal damage in several regions of the brain, including hippocampus. And that is why the ability of the AIDS patients or AIDS survivors to recall memory goes away. Now, the silver lining in all this is this: because of antiretroviral drugs or anti-HIV therapy now, the severity of HIV induced memory damage has gone down. This is because the trafficking which you saw in that animation is not happening anymore. In other words, since anti-HIV drugs are working on the blood cells, the number of replicating virus in the blood has gone down very, very low. But these anti-HIV drugs cannot clear the virus from the brain, and that is why there is some cognitive and motor deficits still in AIDS patients. I hope this answers your question. If not, you can again write to me and we can explain to you. What we also found, as Dr. Basu mentioned, that in India, the strain what we have in HIV C is less severe because the TAT protein is less neurotoxic. Okay, so I think let's, if there are no other further questions.
So I could not see any questions here. Are there any questions that YouTube?
That I am not able to see. So
okay. So if there are no other questions.
We can tell these students that most of students are always welcome. I hope in another 2 months or 3 months we'll be once again go back to our normals in terms of opening the labs. We always welcome students from school, colleges, universities, institutions to come and visit us. The scientists at NBRC have very, very, wonderful work we are doing, and it is, seeing is believing. So once you come and see the human brain, you see us working, you interact with us, you will learn more about it, NBRC as well as neuroscience. So we all will welcome you as soon as the conditions become better.
Okay, with that, if you do not have any other questions, I would like to thank Dr. Seth for this wonderful lucid talk. And also I would like to thank Professor Jain and all other colleagues and students of NBRC for attending this talk, and students from outside. I would request them, if they have any questions, please write to us. Whatever your queries, please feel free to write email to us, we'll respond. And as Dr. Seth mentioned, as soon as the situation will be normal, you can always come to visit NBRC to see the labs and to see the research of your own experience. Thank you so much. Thank you all. Have a good day.
Thank you, Anirban. Thank you all.